Bismillah ar-Rahman ar-Rahim. Good afternoon, and it's my pleasure to moderate this our today's activity from up to 5 o'clock or so. And to start with is a well-known person to you to give us his talk today. Professor or Dr. Altaf. Dr. Altaf is well-known to most of you, if not all. He's a professor and head of the ophthalmic genetic lab at our department at KSU. And he has his PhD from India, and then he did his postdoctoral training in the United States. And he joined us on 2010, now almost 14 years ago or 15 years ago. Yes. And his focus of research is on in our genetic lab. He's the head of the genetic lab in our department. His focus of research is on glaucoma genetics. And nowadays, he's also concentrating on or trying to identify genetic and epigenetic markers or whatever in the field of glaucoma genetics. Dr. Prof, you can have your Altaf, you can have your talk now, and we are willing to listen to you and to hear your talk. Thank you.
Assalamu alaikum and good afternoon, everyone. Thanks for joining me. Thank you, Prof. Saleh, for my kind introduction and for moderating this session. And I'll be talking, as Prof. Saleh mentioned, I'll be talking today about the emerging role of epigenetics in glaucoma, which is a very important and upcoming field in this area. Just to give you a heads up on what I'll be talking about today. So we'll speak briefly. I will briefly touch upon the glaucoma epigenetics. The glaucoma. Thank you. I'm audible right now? Okay. So I will briefly touch upon the glaucoma burden and then the genetic blueprint of glaucoma. Then I will come to the main core of the talk, which covers about the epigenetics, including the DNA methylation and histone modification and microRNA. I'll just give you some examples of the study, which will highlight the role of these markers. And then we'll summarize and conclude the session. So let's begin with glaucoma. So glaucoma is a group of optic neuropathies and the most common cause of irreversible blindness. We are all aware of this. And the main pathological etiologies include elevated IOP, retinal ganglion cell degeneration, and optic nerve cell damage, which eventually leads to loss of vision. While IOP is a major risk of axon damage, damage can also occur at normal IOP, which highlights its complex pathophysiology of glaucoma. So why is glaucoma important? So as you can see from this pie chart, glaucoma has around accounts for almost 12.3% of cases in the eye diseases, which is just lagging behind the most common cataract, but it is more prevalent than other ocular diseases. So again, the high prevalence and the irreversible nature of vision loss makes the research into the pathogenesis of glaucoma and a new treatment absolutely critical. Just coming on the epidemiological relevance. As you can see from this graph, there is an increasing trend of glaucoma prevalence worldwide. A recent meta-analysis showed that number of glaucoma cases in the age group of 40 to 80 has risen from 44 million in 2013 to 65 millions in 2020 and is projected to skyrocket to 112 million by 2040. Again, emphasizing the need for early diagnosis and novel approaches to prevent loss of vision in glaucoma. Coming to what are the common risk factors of glaucoma? They include demographical and geographical reasons like aging, ethnicity, and gender. Then systemic diseases like diabetes and hypertension also cause glaucoma. Then you have well-known ocular factors like IOP, central corneal thickness, and myopia. Then coming to some inherited factors like family history and genetic factors. So what does all this say is like glaucoma is a classic example of complex multifactorial disease. Touching upon the first factor, which is race, age, and ethnicity, this is a graphical representation of how estimated prevalence of POG is impacted by race, age, and ethnicity for men and women combined. The panel A is on the normal scale, and panel B is a logarithmic scale. So if you focus on the panel B, logarithmic scale, you can clearly make out the prevalence of POG is increased in all ages across all the groups, but it varies with geographical and ethnic groups. I've tried to color code these ethnic groups because there are too many colored lines for the ease of for convenience. So you can see the prevalence is highest in Africans across all ages, the blue line. And though Hispanics, it takes a lead above 80 years of age group. The Hispanics and whites show age-dependent increase as compared to the African and Asian ancestries. And men had 33% of higher risk of glaucoma than women across all the age groups. So this ethnic and demographic distribution points to the underlying genetic and possibly an epigenetic susceptibility that are shaped by environmental and lifestyle. Coming to glaucoma genetics, there are a lot of publications and abundant evidence right now to suggest clearly that genetic factors do play a role in glaucoma development. This is true for both inherited familial forms and the common polygenic like POAG, angle closure, and pseudoexfoliation. This forms the molecular basis of investigating glaucoma. As you can see from this table, there are many genes are linked to different subtypes. Over 100 genes are reported to be associated with different types of glaucoma, reflecting the complexity of glaucoma genetics. Glaucoma genetics is thus very heterogeneous, involving structural, regulatory, and metabolic pathways. Although genetic studies have provided a lot of insights into the mechanism, the pathogenesis of glaucoma, these genes do not explain all cases of glaucoma, and the search for other genetic and non-genetic factors is still an active area of investigation. Before moving ahead, let's touch briefly on the heritability of glaucoma. So what is heritability? Heritability, which is denoted by H square, is used to quantify the genetic influence. The value of H square ranges from 0 to 1, which describes what percentage of variability in the trait is due to genetic or environmental factors. Single gene disorders normally have a heritability close to 1, indicating a strong genetic influence. And studies have reported that heritability estimates of POG range between 0.17 to 0.81 and 0.65 for PACG. A heritability value between 0 to 1 is typical for a complex multifactorial trait. So this is not close to 1. It's between that, which indicates that there are presence of other non-genetic influences. So the modern model of glaucoma pathogenesis includes that glaucoma is not just about genes or environment alone, but it is a complex interplay mediated by epigenetic regulations. Epigenetics act as an interface between genetic susceptibility and environmental stresses, modulating disease onset and progression of glaucoma. So what is epigenetics now? Now we come into what is epigenetics. As we all know, DNA is transcribed to RNA, which is then translated to proteins. Now these processes are strongly regulated by epigenetic regulations. In contrast to genetic mechanisms, epigenetic changes refer to the modification of DNA molecule or associated proteins that can alter gene expression without actually changing the underlying DNA sequences. Epigenetic mechanisms have been shown to play a critical role in complex diseases like glaucoma. So, which are these epigenetic regulators? This is again to reiterate how epigenetics could be the missing link between the complex pathogenesis of glaucoma. Environmental factors will interact with genetic factors, and epigenetic will actually regulate them. They can regulate the pathological processes like ECM remodeling, fibrosis, or ROS, reactive oxygen species, TM dysfunctions, IOP, and RGC death, which can lead to glaucoma. So today we'll focus on these epigenetic factors, which is methylation, histone modification, and on-coding RNA. This is just a visual representation of the epigenetic factors, and I will cover about DNA methylation, histone modification, and microRNAs. So let's begin with DNA methylation. So DNA methylation is often referred to as an epigenetic switch. What is methylation basically? So methylation involves adding a methyl group to a cytosine base primarily at the CpG dinucleotides and near the gene promoters. What are the outcomes of these mechanisms? They are very straightforward but powerful. Like it will result in hypermethylation, which is addition of methyl groups, typically leading to gene silencing, which will stop the transcription. Like you will see, when they are hypermethylated, there is no transcription of the gene. And when there is hypomethylation, which refers to removal of the methyl groups, it will lead to gene activation, and there will be transcription leading to formation of the protein. So that's the basic how DNA methylation works. This switch is referred to is normally a reversible switch, which is the fundamental to cellular identity and function. So DNA methylation involves some more key enzymes, which we'll briefly look at. They are referred to as the writers, which are DNA methyl transferases, which actually add the methyl groups. DNMT1s and DNMT3A, 3B, which are involved in maintenance and establishment of new methylation sites. Then there are something called erasers. Erasers are the TET enzymes, 1011 translocation enzymes, which actually control the on and off of the methyl groups. It basically works for removal of the methyl groups. Now, what is the importance for studying the DNA methylation? It is understanding the DNA methylation can provide insights into epigenetic gene regulation. Abnormal DNA patterns have been associated with various diseases, so it has a biomarker as well as therapeutic potential. And as I mentioned, epigenetics which are reversible, so you can actually correct the faulty methylation profile. So it has a promising value in personalized medicine. So let's begin with some examples of evidence in glaucoma, starting with outflow resistance and IOP. So there is this study which was performed in POG patients where blood cells were used. And they reported there was higher amount of global DNA methylation. So global DNA methylation is normally done by either bisulfite sequencing or by ELISA methods are also available nowadays. So what they found was in these patients, there was increased methylation as compared to the controls, suggesting there was the dearrangement or deregulation of the epigenetic control. Then there was some other study which was performed in trabecular meshwork cells. These authors, they reported DNA hypomethylation in POAG and PSAG, which was basically present in the allo repeats. Now, these repeats are involved in genomic stability. So they proposed like increased hypomethylation, which will cause hypomethylation means it will cause activation of the gene. So there is more activation of this gene, which will result in genomic instability and profibrotic phenotype in the TM. They also reported hypermethylation of the HERV K endogenous retrovirus repeat elements. Now these are involved. These are basically involved at embryo developmental stage. So they reported hypermethylation, indicating that there would be local transcriptional regulation contributing to TM dysfunction. So these findings, what they collectively tell us that there is epigenetic changes provide molecular basis for systemic as well as local tissue dysfunction in POAG. Now we come on to a very interesting example where the epigenetic mechanism is targetable. You can actually treat that. So the case of this growth differentiation factor 7, GDF7, which is a member of the TGF beta family, I believe it's a landmark finding. So what the authors reported here was there was an epigenetic dysregulation in the TM cells. They reported a TET-dependent. TET, as I mentioned out earlier, it was the DNA methyl eraser. So they reported there was a TET-dependent GDF promoter hypomethylation. Hypomethylation means it will result in gene activation. So that caused increased production of GDF7. And this GDF7, as you can see in the image, through this BMP receptors and smart signaling will result in overexpression of the profibrotic genes, which will result in fibrosis. And this fibrosis will result in increased outflow resistance and high IOP. Very interestingly was in this study, they provided a therapeutic proof of concept. What they did was they used GDF neutralization antibodies and in a primate model. And they reported that there was reduced TM fibrosis and restored the aqueous flow outflow function. What this study showed was that GDF is a direct, reversible, and targetable epigenetic cause for fibrosis and IOP elevation in POAG and could be a potential novel antifibrotic therapy target. Now, in this pseudo exfoliation, as you all, many will be knowing that LOXL1 is the gene which is normally having a polymorphisms which is involved with pseudoexfoliation glaucoma. Now here these authors reported that LOXL1 promoter was hypermethylated in these tissues which were relevant, lens capsule and human tenon fibroblast. Again, hypermethylation means it causes gene silencing. Gene silencing will cause reduced LOXL1 expression. And since LOXL1, as we know, it is important for ECM cross-linking, reduced expression of LOXL1 would result in defective ECF remodeling, which will lead to accumulation of pseudoexfoliation material, which will block the TM and leading to high IOP. Again, interestingly here, they treated the HTFs with DNMT, which is the DNA methyl transferase inhibitor. They used an inhibitor and they saw that the function of LOXL1 could be restored. So what they showed here was methylation can drive also the classic pathology of pseudoexfoliation by epigenetic silencing of LOXL1 as a molecular switch that dictates the defective ECM remodeling and deposition, and reversal of these epigenetic changes may represent the potential therapeutic target in pseudoexfoliation. Coming to the next part is the example of neurodegeneration, how epigenetics can regulate neurodegeneration with an example in NTG. So in this case, they reported a gene which was modulated, which is a cyclin-dependent kinase inhibitor, CDKN2B. Now, many in this room may be knowing that CDKN2B, there are polymorphisms which are strongly genetically associated with glaucoma in multiple ethnic population. So what the Burden et al., which was, the author did, was they investigated the CDNK methylation status in blood samples of Australian cohort. So they found that this gene promoter was hypermethylated, and they linked it to susceptibility mostly in women population, which was independent of the genetic variant associated with glaucoma. Again, hypermethylation will suppress the activity of CDNK2B, which is actually as a neuroprotective cell cycle regulator. This suppression increases optic nerve. This will increase optic nerve vulnerability and RGC death to non-IOP stresses like vascular dysfunction or oxidative stress. This study highlighted that epigenetic errors through aberrant methylation can impact RGC survival pathways even at normal IOP. Now, this brings us to a very good example. I mean, this is a very good example of how highlighting the therapeutic potential of epigenetic reset using OSK. So in this study, aging, as we all know, is associated with loss of epigenetic information and also loss of retinal ganglion cell function, and which is very similar to what happens in neurodegeneration disease like glaucoma, which is often considered irreversible. So the hypothesis of these studies was, can a partial transient expression of Yamanaka transcription factors, OSK, which stands for this OCT4, SOX4, SOX2 and KELF4, can they reverse the epigenetic age of RGCs and restore youthful function without inducing a cancerous state? The goal was to test if epigenetic reset can heal or regenerate nervous system tissue damaged by age or injury. Just taking a step backward, what are these Yamanaka factors? So Yamanaka factors are the set of 4 transcription factor proteins, OCT4, SOX2, KELF4 and c-MYC, that can turn back the clock on cell's identity. So Yamanaka factors can reprogram the adult somatic cells and turn them back to stem cell-like iPSC state. And this actually forms the basis for regenerative therapies. In theory, what it means, like, if you reprogram the old or damaged cells using these factors, it is possible to reverse cellular aging or repair age-related damage. And in this study, they used this only OSK approach, that is, they omitted the c-MYC oncogene because it is not required for initial cellular reprogramming, and to avoid, since c-MYC is an oncogene, they wanted to avoid the detrimental effects of this gene. Coming back to this study, what did exactly they do for this in vivo epigenetic reprogramming? What they did was they delivered these Yamanaka factors in these AAV viruses directly into the RGCs of a mouse retina. They tested 2 disease models. One was aged mice to assess the reversal of natural age-related vision loss, and second, they use an optic nerve crush model of glaucoma to simulate RGC damage and loss of function as is seen in glaucoma. And they did some functional assessment to measure visual acuity and RGC count. So the findings were phenomenal for this study. What they reported was there was improvement of visual acuity in 12-month-old mice reaching to comparable to young age. I mean, I think if the young age was around 3 to 5 months. So the 12-month age treatment, the mice reverted to the same pattern as for 3 to 5 months age. And also the OSK treatment promoted RGC survival and axon regeneration following the optic nerve crush, which was earlier considered impossible. So the molecular mechanism for this reset was they reversed the DNA methylation aging signatures in the RGCs, effectively making them epigenetically younger. And the recovery of vision was, TET dependent. TET again is a demethylation, if you remember that enzyme, which was actually demethylates. So the recovery of the vision was TET dependent, which was central to DNA demethylation. What they observed was TET inhibition blocked the therapeutic effect of OSK, supporting the functional role of DNA methylation in OSK-induced aging process and vision restoration. Role of other transcription factors and epigenetic modifications, however, was not ruled out. So this study actually provided a proof of concept of how epigenetic reprogramming can actually reverse the aging and also restore vision. But in this, they did a follow-up study. That study was published in Nature, and this again, they did a follow-up study in 2023, which went a little beyond the proof of concept. They followed it for longer duration. The earlier study was only limited for 1 month. Now here they did a longer follow-up. What again they did was, as you can see from this cartoon, they did a gene delivery after 4 weeks. They used a glaucoma model in mouse studies, in mouse model, which was induced via magnetic microbeads. And they did a gene delivery after using this OSK approach. But they used 2 different models for gene a constitutive, which is continuously expressing gene vector, and second they used which was a TET on OSK, which was based on presence of doxycycline in the medium, where they used to give doxycycline to the mice in water, which actually then triggers the OSK expression. So they tested 2 models. One was cyclic OSK, and one was the, as you can see in the green one, the continuous OSK, and with the orange one, which was a cyclic OSK expression. What they observed was there was full vision recovery after 8 weeks within just 2 months by both continuous as well as cyclic OSK treatment. This vision recovery was stable for at least 12 months with continuous expression, with significant vision improvement remaining for 1 month. In the continuous model, it was at least stable for 11 months, and in the cyclic model, after removal of dox from the water, it was stable for only 1 month, but it remained better than the baseline. Interestingly, there was no adverse effect on retina structure or body weight was observed for the continuous expression, providing compelling evidence for efficacy and safety. So what are the conclusions of these, both the studies and implications for epigenetic reset? So what these studies actually suggested were, or demonstrated, transient expression of OSK factors in RGC successfully resets the epigenetic clock, restores the youthful gene expression profile, and enables functional recovery in both aged and damaged neurons. The study represents the first successful demonstration of in vivo neural regeneration achieved through epigenetic reprogramming. The findings of the follow-up study established that epigenetic reprogramming of RGCs is a viable and sustainable approach to regenerate dysfunctional neurons, but they were not dead yet, and recovering loss of vision in glaucoma, and potentially maybe in other age-related disorders. While still in clinical phase, this strategy is more than a proof of concept. These findings solidify epigenetic reprogramming as a viable and sustainable strategy for recovering loss of vision in glaucoma, moving us closer to a future which will complement IOP lowering with direct neural rescue and repair. Coming to another example of using epigenetic clocks, where moving beyond regeneration, we'll see how epigenetic clock can offer prognostic potential. This study hypothesized that mismatch between epigenetic and chronological age could be a risk factor for glaucoma progression. What they did was they recruited a group of POEG patients with faster progression and slow progression and did a DNA methylation profiling. And calculated the epigenetic age using different epigenetic clocks from these blood samples. What they reported was accelerated epigenetic aging was actually associated with 15% higher odds with faster glaucoma progression after adjusting for confounders. Moreover, this relation was strong even in patients with low IOP, indicating that epigenetic aging may predispose the optic nerve to damage from things like oxidative stress even at normal IOP. These findings highlight the potential clinical importance of epigenetic age acceleration as reflected in DNA methylation as a non-invasive predictor of glaucoma progression beyond the influence of established risk factors. However, there is a need for prospective research in this study to establish the causal relationship and potential effects of glaucoma treatments on the results. Now we move on to the second epigenetic mechanism, which is the histone methylation. Now, histone methylation again is a very critical regulatory mechanism, and I've picked up this one example among many others to show how it can play a role in glaucoma. So in this study, the authors investigated large Japanese family with NTG, and using whole exome sequence, they identified a mutation in a MET-L23 gene, which is a methyl transferase like 23 gene, which segregated with the disease phenotype. This gene actually encodes a histone arginine methyltransferase, which is a modifier to control gene excess. This mutation was hypothesized to cause apparent slicing, loss of protein function, disrupting the expression of key neuroprotective genes, rendering RGCs vulnerable to degeneration even at normal IOP. They used a very comprehensive approach. As I mentioned earlier, they used whole exome sequencing to identify the pathogenic mutation. And they also, what they did was they derived induced IPC cells from PBMCs, which were differentiated into RGCs to actually characterize the splicing defect and observe a protein mislocalization in human context. They also created knock-in, knockout mice models of this MET-L23 gene and actually developed the phenotype or the glaucoma phenotype. And also they did molecular assays to actually characterize the downstream histone methylation levels and the inflammatory markers. The major findings of it, they indicated there is a clear epigenetic mechanism involved in this NTG phenotype. MET-L23 actually catalyzes a dimethylation of a histone mark in the retina. This ensures transcription of a neuroprotective gene which negatively regulates NF-kappa B signaling, which is an inflammatory cascade to actually protect the RGCs from glaucomatous damage. This mutation abolishes the MET-L23 activity, leading to a specific loss of function in retina. This loss of function triggers NF-kappa B activation, leading to upregulation of pro-inflammatory cytokines, creating a pro-inflammatory state. Now, this pro-inflammatory state will drive the RGC apoptosis and optic nerve degeneration independent of IOP. This provided a compelling evidence for NTG etiology. What are the conclusions and implications of this study? As I pointed out, they pointed out a very novel etiology for NTG, and they provided the first direct evidence of linking aberrant histone arginine methylation to glaucomatous neurodegeneration. Second, this finding highlighted that NF-kappa B signaling axis and the histone methylation modulators can provide as a promising drug targets to restore epigenetic code by restoring epigenetic code or blocking the downstream inflammation to prevent vision loss. Broader scope will be the study reinforces the crucial role of epigenetics in neurodegenerative diseases. Also demonstrate a concerted role of histone modification and DNA methylation crosstalk in determining gene function and disease susceptibility, shifting the paradigm beyond traditional pressure-based model of glaucoma management. Now we come to the last of our epigenetic mechanisms, which are the microRNAs. So what are microRNAs? MicroRNAs are short non-coding sequences of RNA, which are about 20 to 24 nucleotide in length. They bind to the mRNA targets in the untranslated region and regulate gene expression by promoting transnatural repression or mRNA degradation. There are thousands of these microRNA which are reported in the human genome. Now, these microRNAs are present in most of the ocular fluids and tissues like aqueous humor, serum, and trabecular meshwork, and it has been seen that these microRNA levels are altered significantly, indicating that they do have a regulatory role. What is the importance of microRNAs? MicroRNAs have the exceptional properties as biomarkers. They are remarkably stable across all the fluids under adverse conditions like boiling and long storage. They can be readily detected and have shown to be disease-specific expression patterns, making them ideal candidates for non-invasive liquid biopsies to detect and monitor glaucoma. We'll quickly see to some examples of functional roles of these microRNA that are relevant to glaucoma. Trabecular meshwork is known as it is a critical structure that actually is involved in glaucoma, which regulates the aqueous humor outflow. And in glaucoma pathogenesis, trabecular TM impairment is the primary risk factor for IOP increase. And chronic oxidative stress is a key player in this dysfunction. So in this study, the authors focused on regulation of genes encoding the ECM proteins by miR29 in TM cells. So what they did, they cultured human trabecular meshwork under oxidative stress, and what they found was in oxidative stress there was significant downregulation of this miR29, and in addition there was overexpression of several of the extracellular matrix genes, collagens and all of them, which were known to be regulated by miR29B. This graph shows that how the oxidative stress actually reduced the miR29B levels. You can see they are all downregulated. But what they observed was when you transfect these HTM cells with anti-miR29 to block those things, all these extracellular proteins were downregulated. So transfection with miR29 downregulated all these ECM genes. The conclusion, what the authors concluded, what that M29B negatively regulates expression of multiple genes involved in the synthesis of ECM and TM cells. Downregulation of miR29 contributes to increased expression of several ECM genes, contributing to buildup of ECM material in the TM, increase in outflow resistance and glaucoma risk. And miR29 was reported to be the key modulator of ECM synthesis in human TM cells. Increasing the expression of ECM genes was inhibited, and when you transfect the cells with miR29, as I showed you, the ECM genes were all downregulated, highlighting that therapeutic potential to prevent fibrosis. Second example is of microRNAs where they can regulate IOP. So this is a cluster of miR143 and 145, which is abundantly expressed in trabecular meshwork and smooth muscle cells in IOP, in the eye. This cluster was found to enhance contractility of SMCs in the TM, which is associated with increasing IOP. The study investigated this microRNA cluster as a therapeutic target. In a targeted double knockout deletion of miR143 and 145, they observed that the IOP was considerably reduced by 19%. They also noticed that the outflow facility in these mice was increased by 2-folds. This study suggested that these microRNAs are required for acting cytoskeletal dynamics and contractility in HTM cells, and manipulating these levels may reduce IOP and increase outflow, and thus can have a therapeutic implication in glaucoma. Next example we'll see is about this microRNA 100 and 96, which are involved in RGC apoptosis. Oxidative stress, again, the authors, what they did was they used this RGC5 cells. They treated with H2O2 to create the oxidative stress. And under this oxidative stress, miR100 levels were actually upregulated, and they observed apoptosis in a dose-dependent manner. If you can see on the graph on the left side, there is increase in the miR100 levels as the concentration of under increased oxidative stress. And in the right hand side, you can see there is increased apoptosis. What they observed was when they inhibited this miR100 using miR inhibitors, it protected against oxidative stress and promoted neural nerve growth through phosphorylation pathways. You can see on this graph on the left hand side, treatment with miR inhibitors reduced the apoptosis and increased the nerve length. What the authors concluded was that the functional role of miR100 in retina, and they suggested that it could be associated with glaucoma pathogenesis in which RGCs were preferentially damaged. In another study, they evaluated miR96. They saw that miR96 was significantly reduced in these RGC5 cells and induced apoptosis through activation of caspases. Caspase 2 was confirmed to be the direct target of MIR96, and silencing of this caspase 2 rescued the apoptosis in RGC5 cells induced by upregulation of MIR96. MIR96 was reputed to be the important regulator of RGC5 cell survival and apoptosis, possibly via caspase 2. Now, after functional, we'll quickly look at some differential expression studies of microRNA that was done to evaluate this role as a biomarker. In this study, the authors investigated a small group of POG and pseudoexfoliation from aqueous humor for microRNA. And what they observed was, they observed a panel of microRNA that were altered in POG group. Then they altered that there is a panel of microRNA that were altered in the pseudoexfoliation group. And there was microRNA which was very significant to POG group only. What it showed was that microRNA profiles can be specific for disease-specific subtypes. And they could also aid in differential diagnosis between POG and pseudoexfoliation glaucoma. There were, in addition to aqueous humors, there were other circulating microRNA studies that were performed in peripheral blood samples and tear samples to investigate if circulating microRNA can serve more for non-invasive testing. In this study, Rao et al. Investigated, again in a small group, they investigated pseudoexfoliation and ocular hypertensive patients where they reported 12 microRNAs that were differentially expressed. And these microRNA pathway enrichment showed that most of these microRNAs were actually targeting TGF, ECM, remodeling, and inflammation, which are actually core pathways that are implicated in glaucoma pathogenesis. Identification, then there is another study where they compared both plasma and aqueous humor samples in a small group of patients and showed that a combination of microRNAs, they observed that there were three microRNAs which showed an AUC of 0.91 with high sensitivity and specificity. And 0.91 is a strong indicator of the diagnostic potential of these microRNAs. What they suggested was screening for a panel of microRNAs would have more diagnostic relevance. Then lastly, there are also there are studies which evaluated tears as another promising non-invasive source. These studies successfully profiled microRNA signature from tear samples in ocular hypertension using next generation sequencing. They identified a panel of 8 microRNAs between POAG and ocular hypertension. And this was the first study to identify microRNAs from individuals with ocular hypertension and glaucoma patient, revealing its potential as a molecular genetic biomarker for glaucoma subjects. They observed some AOC for some of these were more than 0.75, indicating that these microRNAs could serve as a good predictor for POAG. Again, they did some pathway analysis and observed that all the glaucoma-related pathways were associated with this microRNA function. So this is another example coming back to NTG. This study evaluated microRNA in aqueous humor of NTG patients. And what they observed was there are 8 microRNAs that were dysregulated in these NTG patients. And most of them were associated with apoptosis, autophagy, neurogenesis, supporting the idea that NTG has having a primary new generated component which are driven by epigenetic changes. To summarize, so far what I presented was to indicate that how epigenetics can have a mechanistic view also, plus they can have a biomarker potential also, plus a therapeutic approach as well, target as well. So to summarize what we looked at today, first was DNA methylation. As I mentioned, it works as a reversible switch for gene silencing and gene activation, where these can work in TM, the retina, and aging and glaucoma. We saw different examples how GDF7 can lead to fibrosis, CDK and 2B can increase RGC vulnerability, and how epigenetic resetting can be involved using these OSK factors. We looked at histone modification through this example of METL23 mutation in NTG. Then we saw some role of a microRNA in the TM in RGCs and how they can serve as biomarkers in aqueous humor and plasma samples or tear samples. So what are the translation and clinical potential of these all these evidences? They have a diagnostic potential. Blood-based epigenetic clocks, what we looked at, can serve as predictor for glaucoma progression. Methylation signatures can serve as a non-invasive marker for detection. MicroRNA panels in aqueous humor or tear samples of plasma can allow subtype discrimination. So epigenetic clock, DNA methylation, and microRNA panel can serve as non-invasive biomarkers. They have a therapeutic potential as well. We saw that the epigenetic can be reversed using OSK approach. So it indicates that epigenetic resetting can be done. GDF neutralization, you can actually control some of the genes through your antibodies to reduce fibrosis and IOP in IOP models. We can develop a DNMT, a DNA methyltransferases or TET modulators or microRNA mimics that can be developed. So all these approaches serve as a good drug target for in these studies. And of course, the main ultimate goal will be to move toward personalized care. So we can integrate both genetic and epigenetic profiling. Subtypes, specific biomarkers can guide treatment choices. Epigenetic therapies can be futuristically complement IOP lowering strategies. So methylation profiling can actually serve as guiding specific subtypes treatment. What is the future direction we can go from these studies? We need to perform, as I said, these are all in preclinical stages and most of them were performed in small sample size. So definitely we need large cohort studies for all these biomarkers for APH, be it microRNA panel or GDF5 methylation studies. Then a step ahead would be to do using gene editing techniques for these genes to actually see what is happening. And then of course this OSK approach, as it was done in mouse model, so you need to validate them in large animal models. Then the final goal will be the clinical translation, will be to integrate all these into clinical guidelines and develop risk stratification models and combining the traditional and epigenetic factors. Just to conclude, epigenetics is transforming our understanding of glaucoma from static pressure disease to a dynamic modifiable interface between genes and environment, offering new tools for early detection, targeted therapy, and vision preservation. Thank you. Thank you for your attention.